Welcome and thank you all for attending. I am Elizabeth Mansfield and I am a research chemist at the National Institute of Standards and Technology. It is my privilege to host this meeting on behalf of the National Nanotechnology Initiative, or the NNI. The NNI is a U.S. government R&D initiative consisting of over 20 federal agencies and departments who work together to achieve the shared vision of a future in which the ability to understand and control matter at the nanoscale leads to ongoing revolutions in technology and industry that benefit society. Over the past 20 years, R&D, nearly all of the areas of research that require the development of novel tools and methods and techniques to characterize and evaluate materials, devices, and formulation have required metrology. Nanometrology, the study of nanoscale measurement, presents a unique set of challenges due to the small size of materials, often requiring more sensitive and innovative tools, methods, and techniques to obtain and understand characterization data. The Nanometrology Webinar Series is to highlight the challenges, advancements, and overall importance of measurement technologies that help researchers gain information about nanomaterials and systems to generate improved data, experimental design, and innovation. The series began with an introduction to nanometrology in January, followed by a webinar on nanometrology for food, agriculture, and the environment. And finally, a webinar in March on metrology of nanoscale medical and pharmaceutical products. Recordings of these webinars have been posted to the nano.gov website. The final webinar in this series is today, April 5th, on metrology of nanoparticles in electronics. This webinar will address recent advances and current measurement challenges in the synthesis, characterization, and application of nanoparticles in electronics. We are lucky to have with us today Nicholas Boris. Nick Boris is an associate professor in the Department of Physics at Montana State University and the associate director of Monarch, NSF Quantum Foundry. Nick earned his Bachelor of Science degree at the Colorado School of Mines in Mathematics and Computer Science. Nick earned his PhD in physics from the University of Utah, working in the optoelectronic properties of nanoscale metal and semiconductor systems. He spent time at the Molecular Foundry at Lawrence Berkeley National Lab, where he led multiple studies of nanoscale optoelectronic properties of 2D semiconductors before he moved to Montana State University to start a research group that is focused on exploring quantum phenomena in low-dimensional material systems with nano-optical imaging and spectroscopic techniques in both the steady state and transitional and transient temporal domains. Among others, a current major research direction in the Boris Lab is to understand how local strain, material composition, and heterostructure configuration affect multi-exciton phenomena in 2D semiconductors, with a particular emphasis on how the phenomena can be enhanced and ultimately utilized for novel, non-classical light sources in quantum photonic device architectures. Our second speaker will be Dr. Farnaz Niroui, an associate professor in electrical engineering and computer science at the Massachusetts Institute of Technology. Her research pushes the limits of nanoscale engineering to develop new paradigms for active nanoscale devices and systems for diverse emerging applications in electronics and optoelectronics. Prior to MIT, Farnaz was a Miller postdoctoral fellow at the University of California, Berkeley. She received her PhD in electrical engineering from MIT and completed her undergraduate studies in the nanotechnology engineering at the University of Waterloo. Farnaz has been the recipient of awards including the DARPA Young Faculty Award, NSF Career Award, MIT EECS Outstanding Educator Award, and the Miller Research Fellowship. Our final speaker today will be Dr. Emery Chan, who is a staff scientist at the Molecular Foundry, a Department of Energy Nanoscience user facility at Lawrence Berkeley National Lab. Dr. Chan received his bachelor's in chemistry at Stanford University and his PhD in physical chemistry at the University of California in Berkeley. Dr. Chan's research includes high throughput synthesis and characterization of materials, in situ liquid cell TEM, and developing optical applications that harness the complex energy transfer networks in lanthanide-doped nanomaterials. To support these research thrusts, Dr. Chan has developed robotic workflows that increase the reproducibility and throughput of colloid nanoparticle synthesis, while also enabling machine learning guided experimentation. These capabilities are offered to scientists worldwide as part of the Molecular Foundries user program. Some housekeeping before we jump into the first presentation. This meeting will be recorded and posted later online. Please use the question and answer chat to ask questions. Questions will be answered at the end of the webinar in the Q&A session. I will now hand it over to Nick Boris for his presentation.
All right. Thank you very much, Elizabeth, for the introduction, and thanks to the organizers for inviting me to participate in the webinar. I mean, it's really amazing to be, kind of co-listed with Emery as well as Faraz. So thank you very much. I trust that everything's functioning fine with the share.
Yep, looks good.
Excellent, thank you. All right, yeah, so as Liv said, I'm Nick Boris. I'm coming from Montana State University. I'm an associate professor in the Department of Physics there, and I am also the associate director of what is called the Monarch NSF Quantum Foundry. And so my goal today in sort of the next 15 minutes is to give a little bit of an overview of nano-optical characterization applied to 2D material systems, and how this technique kind of provides us with the opportunity to gain a lot of information about these material systems for applications not only in electronics, but photonics, as well as quantum technologies. And so 2D materials are kind of a focus. So I just kind of want to provide a gentle kind of introduction into what two-dimensional materials are, or what I am referring to when I talk about two-dimensional materials. And so this figure right here is an example of two-dimensional materials. So depending on kind of estimates, there's about 1,200 known 2D materials that kind of fit into this category. They come as superconductors, charge density wave materials, magnetic materials, semiconductors, ferroelectrics, exotonic insulators, topological materials, and so on. And the unifying property of all these bulk crystals that you see here is that they consist of atomic sheets, maybe 1 atomic layer thick, 2 atomic or 3 atomic layers thick, that are stacked on top of one another. And so the chemical bonding within the individual sheets is typically very strong, whereas the bonding from sheet to sheet, or the interlayer bonding, is significantly weaker. And so what this enables researchers to do, and the researchers really across the United States and across the world, is to quite literally peel individual layers off of these crystals and isolate these true two-dimensional atomic systems. And so it's really remarkable is that we can study all of these kind of effects in 2D, but then what is even more remarkable and more exciting is that we can then kind of pick these individual atomic sheets up and stack them on top of one another. And so we can, for instance, combine, magnetic phenomena in one 2D material with semiconducting phenomena in another 2D material. And so these material systems, have really led to a large number of exciting studies and advances in condensed matter physics, where you can kind of study phenomena in truly a 2D limit. So in the semiconductor systems, there's been a lot of intense study of the exotonic and excited states in 2D semiconductors, and how they go from excitons from a 3D system into a 2D system, and the emergent phenomena. There's also been a significant effort in studying superconductivity as well as charge density wave phenomena in these two-dimensional materials. Ferromagnetism in the 2D limit. So this was discovered and reported back in 2017. And then also there is a large variety of studies of transport phenomena, things like the quantum Hall effect, the Hall effect that you can study in this 2D limit. So from a condensed matter point, very interesting. But things really get exciting when you start thinking about what has been achieved with heterostructures when we combine different two-dimensional materials together. And so kind of, I'd say probably the most famous example of this is the realization of superconductivity in twisted bilayer graphene. So this was 2 sheets of graphene stacked together with a relative twist in between the individual sheets, of about 1.1 degrees. Right. You can also use this to realize what are known as Wigner crystals in twisted 2D semiconductors, as well as engineer excitons in 2D semiconductors and really control kind of the configuration of the constituent negatively charged particle and positively charged particle with these systems. And furthermore, use kind of this twist degree of freedom or the ability to twist individual layers with one another to create kind of more exotic kind of interlayer excitons that are trapped in what are known as a Moiré potential. And so I guess, in 15 minutes, I can't possibly explain all the details here, but the take-home message is that really when we start talking about the physics in these material systems, which are all very exciting, this really constitutes a convergence of, nanoscale structure and properties of the 2D materials with many body phenomena and exotic emergent phenomena in condensed matter physics. And so with that said, nanoscale characterization of these systems is really important. And so, one commonly applied technique is scanning tunneling microscopy and STM inspired techniques to image kind of the nanoscale structure, and then also AFM-based characterization. And so this is a really great example of a open question, or still a question that's being actively studied in this field, is when you take two-dimensional lattices and stack them with a relative twist, if the lattices are completely rigid, you're expected to form this Moiré pattern. However, in reality, the lattices aren't strictly rigid, and they can reconstruct. And rather than a Moiré pattern, you kind of get these patterns. And so the big question is how do these nanoscale properties affect some of the device characteristics and emergent phenomena that has been observed in the past? And so complementing things like scanning tunneling microscopy and AFM-based techniques is optical techniques, which really provide kind of an added dimension into characterization and give you access to excited states. And so, optical microscopy is really powerful for studying kind of excited state phenomena in many different material systems. You can study excitation processes from the ground state into a variety of higher energy states. You can study the relaxation processes amongst the higher energy excited states, as well as radiative relaxation processes. And so this is great. I mean, really powerful characterization, but there's a fundamental problem, is that the spatial resolution of a conventional or normal optical microscope system is limited by the diffraction limit to about 500 nanometers. And so this is an example of light emission from a 2D semiconductor taken on an optical microscope. Color scale indicates brightness. Scale bar here is about 1 micron. And you can see, you can map it out, but there's not a lot of interesting features. And so this resolution limit, doesn't really match to the critical length scales of many processes in 2D materials. And so this is one of my common slides. It's kind of oriented towards the physics in 2D semiconductors, but it applies also to all the other systems. So the critical length scales of things like variations in carrier density, strain fields, interfaces, localization effects, and Moiré potentials, as well as domains and grain boundaries, kind of have these critical length scale below this diffraction limit. And so the good news is that there's a way to bridge this resolution gap, and this is with nano-optical imaging and spectroscopy. And so this is a schematic of tip-enhanced optical microscopy. And so rather than using just a normal optical microscope, what this technique involves is bringing a sharp gold tip, usually a metal-plated AFM probe, in very close proximity to the sample. And then this tip sample junction is illuminated with an incident laser. This incident laser causes the conduction electrons in the tip to oscillate at the same frequency as the incident laser. And the net result is you get a concentration of localized oscillating electric fields right at the apex of the tip. And these fields are confined to length scales determined by the radius of curvature of the tip, which can get down to about 20 nanometers in space. And so the end result is that you can use this tip-enhanced configuration, and you use these oscillating electric fields at the tip as a local light source to perform optical microscopy and spectroscopy. And so there's a lot of different modalities of this. There's kind of the aperture-based near-field optical microscopy, scattering-based, as well as then the scattering base with a metallized tip for doing tip-enhanced Raman and tip-enhanced photoluminescence. And so I won't go into great detail on all these different modalities and just kind of refer you to this review article for further information. But just to kind of give you an idea of what can be achieved with this, so here's this image of this two-dimensional crystallite, the light emission taken on the normal confocal microphotoluminescence setup with a resolution somewhere between 1 micron and 500 nanometers. And here's an image of that same crystallite taken with a nano-optical probe that has a resolution on order of 50 nanometers. And you can see that a really compelling amount of disorder and localized states kind of emerge kind of when you get down into this much smaller spatial resolutions. And so my group, along with collaborators, Jim Shuck, who's now at Columbia, Andrei Krayev at Hariba, and Patrick Kung at the University of Alabama, we've used this to study a lot of nanoscale phenomena in 2D semiconductors. So we started looking at edge states as well as interior disorder due to carrier variations in 2D semiconductors. We've also used this to study the interfaces that form in lateral heterostructures of, say, two semiconductors. So in this case, single layer tungsten disulfide and single layer molybdenum disulfide. And then, most recently, we've been using this technique to study strain localized excitons in 2D semiconductors and really get an idea of how strain on the nanoscale, so on order of 100 nanometers, creates localized potentials that can be used for quantum information science and technology applications. And so, we're part of a much broader community. And so, I sat down over, Wednesday of this week and kind of tried to form a list of all the groups that I know who are actively working in this area and making strong contributions. You can kind of assume that I probably have a hit rate here of about 50%. So I'm guessing that this, we have probably 30, maybe 50 active groups in this. People doing some really nice work. And so a group out of Brazil has done tip-enhanced Raman scattering of twisted bilayer graphene to reveal the kind of the moire potential that emerges. We have the Park and Rashka groups have looked at a lot of in-plane strain and strain effects using tip-enhanced spectroscopy. And then there's a whole new kind of emerging field, which we could kind of attribute to Dmitry Bazov, Josh Caldwell, and the Fay group, where you look at surface-confined propagating hybrid light matter wave states in 2D materials, states called polaritons. And this tip-enhanced spectroscopy technique is a really great way to image and demonstrate that these polaritons exist, but then also map out their dispersion relationships. And then finally, there's several groups who are really working on the chemistry and quantum effects that occur in kind of the tip sample junction when you bring this metal tip in really close proximity to a material and drive it pretty intensely with light. And so I apologize if I've missed anybody out there. It's certainly not intentional. But yeah, so it's a very vibrant field, and I feel really fortunate to be part of it. So you might notice a little bit, and so just in my last kind of 2 minutes here, is that there's a little bit of a mismatch between a lot of these examples that I've shown and sort of the complicated systems that I introduced in 2D materials. And so that kind of gives rise to some opportunities and challenges that I'd kind of like to conclude with and maybe provide some outlooks here. And so most of these measurements, not necessarily all of them, but most of them occur under ambient conditions and room temperature conditions. In particular for quantum materials development, we really like to push these techniques to cryogenic and ultra high vacuum environments. And then there's a really a big challenge in building 2D material systems that have like this really like high quality emergent phenomena that are compatible with nano-optical techniques. And so that has kind of motivated a little bit of the work that we have going on at the Monarch Quantum Foundry. And so we're a joint program between Montana State University and University of Arkansas to, broadly speaking, accelerate 2D materials research to solve challenges in quantum information science and technology. We have a couple of main thrusts. And what I just want to focus on today is some of our infrastructure that is focused on tackling these challenges. And so The first bit is nano-optical characterization at low temperature. And so we started a project with Renaissance Scientific and 49 Design to build a nano-optical microscope that has a 111 vector magnet, as well as can operate at a base temperature of about 2 Kelvin. So we're really looking to be able to do all these nano-optical measurements at 4 Kelvin and in an external magnetic field. And then also, we're working on the fabrication challenges of going from these bulk crystals of 2D materials to devices, particularly for nano-optical applications. And so to that end, we're building 2D quantum, what we call the 2D quantum materials pipelines. And so these are largely inspired by the work of Emery and similar groups. And the idea is to build robotic elements that speed up this process and tackle kind of the 3 main processes: exfoliation, optical identification, and heterostructure stacking. And so we're making good progress on these. And so just kind of an example of some of these robotic elements that are currently under development. Don't have so much time. I can kind of see that I'm running out of time. So I'm going to kind of wrap it up there and just kind of conclude with this slide, more information. Happy to provide more information about Monarch. And then also I figured I'd conclude with a couple of review papers in near field imaging that were recently published. And so thank you.
Thank you, Nick. Up next for a presentation is Dr. Farnaz Neroui from MIT.
Thank you, Elizabeth, for the introduction and also for having me as part of this panel. So what I'm going to do is spend the next 15 minutes to try to give you some idea of the prospects of device engineering with nanoparticle building blocks down to a single particle resolution. And along the way, I'm going to highlight challenges and also opportunities in metrology. We're all very familiar that nanotechnology has had tremendous technological and scientific impact over the past several decades. Many applications of nanotechnology, though, commonly focus on dimensions that are tens of nanometers in size and often much larger, leaving behind smaller dimension less explored. And that in part is due to challenges in nanoscale processing as well as metrology as we scale down to even smaller dimensions. With the rapid advancements in artificial intelligence, Internet of Things, quantum technology, so new emerging technologies are needed, which would require us to be able to push the limits of nanoscale engineering towards even smaller dimensions, down towards the atomic scale. If we can do that, then we have access to the unique physics that occurs at these smaller dimensions, plus the multitude of new and improved functionalities that the rapidly growing library of nanomaterials can offer us. Nanomaterials like two-dimensional materials, just Nick talk about colloidal nanoparticles, generally low-dimensional materials, molecular materials, and nanostructured matter. By doing so, then we can develop new platforms of active nanoscale devices and systems, and that's the focus of my research group. What I'm going to focus on today is largely about colloidal nanoparticles and using them as nanoscale building blocks and devices. And colloidal nanoparticles are appealing because through chemical synthesis, they can have finely tuned structure, shape, and composition in such a way that they can gain access to diverse functionalities, ranging from metallic, insulating, semiconducting, magnetic to others. And in many cases, these functionalities and properties are superior or unique to what we can find in their bulk counterparts. Now, nanoparticles have been broadly used in diverse areas of developing electronic, optoelectronic devices. For example, as thin films, they've been used in thin film solar cells or light emitting diodes, as well as in composite form for a variety of different types of sensors, and in some cases, even at the single particle level to develop devices, for example, for plasmonics. And what my group's interest is be able to gain control down to the single nanoparticle level to be able to have integrated nano devices using unique properties of colloidal nanoparticles. Now you can imagine that is a challenging task. Nanoparticles are made in solution, so they're stochastic. Here I'm showing you as an example, just a cluster of gold nanocubes that have been colloidally colloidally synthesized. So if we have them on a substrate at low density, you can see how the particles are stochastic. And what I'm showing you here is a dark field image where individual red dots correspond to an individual nanocube, emphasizing that dark field imaging actually provides us with a high resolution yet high throughput imaging approach to be able to characterize distribution of these nanoparticles. So colloidal nanoparticles on surface are stochastic. Integrated devices required to be deterministic, scalable as well, give us diverse access to fabricating a variety of different types of functional heterostructures. Heterostructures are core to any nanoscale device design. So it becomes important for us to be able to come up with nanoscale processing techniques that give us the level of control and resolution at the individual nanoparticle level, but at the same time, new metrology techniques to be able to visualize and understand what is exactly happening to the nanoparticle as it gets integrated into the device. Another point to keep in mind, as the nanoparticles get incorporated into the device, which is either electrically, optically, mechanically, or through other means active, the properties of nanoparticle is now going to start being influenced by the complex environment surrounding it. So oftentimes conventional techniques, SEM, TEM, AFM wouldn't be sufficient for studying the nanoparticle in a device format. So other techniques would be necessary. So in one approach, what we've developed is nanoparticle contact printing. I'm describing the approach to you here using gold nanocubes as an example material. So in this approach, first we use capillary assembly to assemble the nanoparticles onto a topographical template that is lithographically defined. I'm showing you here an array of 50 by 50 nanocubes. This is a dark field image of spatially arranged nanoparticles. So at this point, the nanoparticles are out of the solution, and the inset shows AFM characterization of the particles, and that gives you a more clear information about the structure of the nanocubes and exact placement. Now, at this point, the nanoparticles are actually embedded into the template material that you've used to be able to spatially arrange the particles. And when it comes to nanoscale devices, surfaces and interfaces need to be accessible to be able to incorporate them into a device. So what we've done is add an additional step where we can now take the assembled particles and without needing any sort of treatment to the surface, sacrificial layers, or high temperatures, transfer print them onto arbitrary surfaces with high yield. And we do that by engineering of van der Waals forces between the particle and the template, as well as the particles and the receiving substrate. So in this case, we take the nanoparticles that are assembled onto a PDMS template, and you can see they're printed onto a silicon surface with more than 95% yield. And again, in this case, a combination of TM, AFM, and more high throughput optical and dark field imaging has been used to be able to characterize the steps. And now that we have an array of these particles, we can do high throughput characterization of metrics that are important for processing development. For example, placement accuracy. So in this case, we characterize over more than 13,000 particles, placement accuracy of less than 50 nanometer. As we have control over placement of the nanoparticles, we can also achieve more complex structures and also be able to process the nanoparticles onto arbitrary surfaces. Showing you here an example of the nanoparticle on gold, metallic surface, dielectric surface, semiconducting surface, as well as a flexible polymer. The approach is also compatible with diverse types of nanoparticles beyond gold nanocubes to nanoparticles of other composition, as well as shape, even a sphere, which has a very limited contact area to the underlying substrate. Now, when it comes to nanoscale processing of extreme nanoscale devices, it becomes very important for us to make sure surfaces and interfaces are pristine. And that's something difficult to characterize through conventional techniques, in particular, when the surface and interface is embedded into the device. So for us to be able to characterize the pristine nature of our process, we used our process to fabricate these nanoparticle on mere plasmonic cavities, which allow field enhancement, and as a result, Raman field concentration, and as a result, Raman enhancement of any material that is placed in the gap. We use this Raman enhancement then to be able to probe the material on the surface, and as a result be able to make a conclusion about whether our processing has introduced any damage to the surface. As an example, we incorporated monolayers of biphenylthiol as well as monolayers of 2D materials into these junctions. And in particular, we choose these materials because their atomic nature oftentimes make them quite delicate and sensitive to any sort of other conventional processing technique. So it would be a good material system for us to probe the capabilities of our approach. So in this case, once we incorporate the materials into devices made through our approach using individual nanoparticle plasmonic cavities and perform enhanced Raman spectroscopy, we see through the Raman signature that the Raman signature of the materials is exactly the same as the pristine material, suggesting that there is no additional damage that's introduced to the material due to the processing technique. It is also important for nanoscale devices that are made out of nanoparticles to actually have a uniform performance across an array of devices. So in this case, our approach allows us to create large arrays of devices. Over 2,000 devices be characterized through dark field scattering spectroscopy, and you can see the optical response is quite consistent. Now compare this against devices that would have been made conventionally, and with conventionally the most common technique of making such devices out of colloidal materials is just drop casting them. So you can see that our approach is way more uniform compared to the stochastic nature of the conventional processing, making it more suited for practical device integration. And that's actually intentionally built into our processing. Our capillary assembly step is designed so that it allows us to be able to do shape filtering of byproducts of chemical synthesis, as well as do size filtering. So another side point I would like to mention here, even though the approach is being developed to enable device integration of the nanoparticles, but the approach itself could actually contribute to new paths towards high throughput metrology. For example, creating large area arrays of nano materials on TM samples to be able to enable high throughput TM studies. To show the integration capability of our approach, we integrated our plasmonic cavities with molecular emitters, designing the cavities to have resonance that overlaps with the absorption and emission spectra of the particular molecule that we're using here. Here's an example of an array of such devices. You can see through this scattering measurement of an array of 5 by 5 of the devices that the response is quite consistent and reproducible and uniform through use of fluorescence imaging over a large area. Then we can see that the individual devices give fluorescence enhancement and characterize that to be on the order of 123 volts. Now, the technique also allows us to do correlated studies. So the same 5 by 5 array of devices that we've measured here in terms of scattering response, we can then use to do fluorescence spectroscopy, and in particular measure their decay time, showing about 77 times enhancement of spontaneous emission rate. So a point to keep in mind here, such techniques, in addition to having contributions to device fabrication and enabling new device platforms, again, introduce a new path for us to be able to do correlated multimodal spectroscopy. And oftentimes multimodal spectroscopy is necessary at such scale to be able to best understand the effect of multi-physics on the performance of the devices. So far I told you about the possibility of using a direct assembly and contact printing approach to incorporate nanoparticles into functional devices. An alternative possible approach is to actually make the nanocrystals locally on site where you need them to be incorporated into the device. I'll give you an example of the platform we've developed based on metal halide perovskites. So halide perovskites are a growing class of materials that have found quite a lot of applications in thin film solar cells, LEDs, lasers, photodetectors, amongst others. Many applications of perovskite devices, though, have been limited to micron scale devices, smallest, many cases, thin film macro scale devices, despite the promise that the superior optoelectronic properties would offer for nanoscale devices if their integration was not challenged by the processing limitation. So in the approach that we've developed, I won't have time to go through the details, but through changing the surface topography and having asymmetric wettability, we can actually localize the chemical reaction to nanoscale sites over wafer scale. And that allows us to be able to hit arrays of these precisely defined nanocrystals, in this case, showing you an SEM of cesium lead bromide. Using SEM as well as EDX, we can characterize the structural detail as well as the composition of these materials, just to confirm we have the material that we meant to make. As well, using optical spectroscopy, then we can have an approach for characterizing the material further, both independently and afterwards when integrated into a device. Using sub-cross-section imaging under TEM, then we can characterize the crystal structure of the particle that we have, and in this case, confirm that the particles formed are actually single crystalline. Again, the process is scalable, so suited for device integration. So we also need more high throughput and larger area characterization techniques, which are often optical in nature. A unique capability that our approach offers is that we can systematically change processing condition so that we can see the effect on the intrinsic property of the nanomaterials. In this case, I'm showing you we can systematically change the size of the nanocrystals that are formed on chip, which corresponds directly to the shift in their optical response. Such an approach would be highly important for us to be able to do high throughput scientific discovery of these emerging materials, as well as be able to incorporate them into device platforms. I'll end with the last two slides, where our approach actually now allows us to then take these individual nanocrystals to incorporate them into more complex devices. Here showing you the first demonstration of deterministically formed nano LEDs out of individual crystals of halide perovskites, in particular cesium lead bromide, opening up many opportunities for use of these high density on-chip integrated LEDs. So just to end, I hope I was able to give you an idea of the prospects of being able to achieve nanoscale control over individual nanoparticles for their integration into emerging nanoscale devices. Along the way, of course, there are going to be processing challenges that have to be addressed, as well as metrology challenges. But the point to keep in mind is that the sort of processing that I told you about also offer new metrology platforms, whether it is for high resolution studies of nanoscale processes that would otherwise be hard to probe, or providing platforms for high throughput correlate and multimodal metrology of these emerging nanomaterials. And just thanking my students and the funding agencies, and thank you for your time.
Thank you. Our next presentation will be Dr. Emery Chan from Lawrence Berkeley National Laboratory. Looks good. Thank you.
All right, good morning. My name is Emery Chan. I'm a staff scientist at the Molecular Foundry at Lawrence Berkeley National Laboratory. And today I'm going to tell you about our work using robots to synthesize and characterize nanoparticles. So our center is located right above UC Berkeley in the San Francisco Bay Area. And the Foundry is a DOE nanoscale user facility. So that means that anyone from around the world can come use one of our 7 facilities to synthesize nanomaterials, to characterize them, which is of course important because you need to know what you made, integrate them into different devices, and also understand them using theory. So I work in the inorganic facility at the foundry, and I make colloidal nanoparticles. So Farnaz gave a really beautiful introduction to nanoparticles, so I don't have to go into too much detail. But I also work on colloidal nanoparticles, and that means they are suspended in solution. And there's a lot of advantages for that, in particular, being able to process them using liquids and to paint them onto surfaces rather than having to deal with big chunky crystals. So we study these nanoparticles mainly because they exhibit size-dependent physical properties. And the most salient example of that is their change in color as they change their size due to various quantum confinement effects. So, of course, this won the Nobel Prize last year, and we're still amazed because these particles have been around long enough that they're actually in commercial products. So because they glow pretty colors, you can use them for lighting and displays. So you can go to the store and buy a Samsung QLED TV that has a really high color gamut. You can also use them to label different parts of a cell so that they glow different colors. But beyond these optical applications, there are other size-dependent properties. For example, some metal nanoparticles have higher reactivities or can be antibacterials. And even if these materials don't have size-dependent properties, the fact that you can process them in solution makes them useful for coatings, for printing, for example, circuits or 3D printing, and even using them for sunscreen and cosmetics. So you need to make really reproducible nanoparticles with reproducible structures and properties, but this is complicated because nanoparticles themselves are more complicated than you would think. For example, they have compositional complexity, structural complexity, because they may have heterostructuring, and also interfacial complexity, because when you need to use them for these applications such as biolabeling, you need to put ligands on their surface and even polymer coat them, and then have different kinds of chemical functionality for linking them to whatever you're trying to bind to. So on top of that, the synthesis is fairly complicated. It's not one of these syntheses where you start with a molecule and you go to another molecule and you're done, right? Instead, there are many different pathways that can produce different products, and they're essentially a very complex reaction network. And so to optimize this network, you need to iterate through all the different permutations of this network and see how you can get the one thing that you really want. So on top of that, you need to find the right formulation parameters, such as precursors, and the right process parameters, such as reaction time. So to develop the ideal nanocrystal synthesis, there are literally millions of combinations, and no human can actually test all of those possible combinations. So recently, there's been a big push to use robots and machine learning to guide materials discovery. So for example, this work by Andy Cooper using robotic chemists has really inspired many people. Also, there's been many mandates from the various DOE and other funding agencies in their workshop reports for utilizing, for example, machine learning and automation to advance synthesis science. So this is a relatively recent phenomenon, but we've actually been working on the automated synthesis of nanoparticles for over 15 years. So in my postdoc at the Foundry, I developed this robot called Wanda, which stands for the Workstation for Automated Nanomaterials Discovery and Analysis. And this robot can do most of the things that a graduate student can do. So for example, you can see right now this robotic arm is weighing things. And let me know if you can't see the video. So weighing these aliquots that it takes from our reactions is really important, because when you're trying to characterize them, you may need to normalize by the amount of materials that you took from the reaction. So to step back a little bit, the reactions are performed in these wells of microplates or in this larger reactor. And so pipettes on the arms of these robots can slurp up reactions, precursors, inject them into a vial, heat and stir that vial, and then you have your nanoparticles. So the secret sauce, or the really unique capability of Wanda, is this low thermal mass reactor. So these quantum dots that we talk about, so semiconductor nanoparticles, are made at very high temperatures. So you normally can't just buy an off-the-shelf robot that can go up to these temperatures. So we custom designed this reactor that can be heated up very quickly and also cooled very quickly. And that's really important because nanoparticles don't stop growing when you get to the size that you want them to grow to. You have to really shut down that reaction very quickly. And in this case, we use a shroud of nitrogen gas that quickly cools this vial. So of course, if you can make a lot of nanoparticles, you need to characterize them very quickly, as the previous talk mentions. So we try to use optical methods like absorption and fluorescence because they're non-invasive. They usually allow you to perform them without purifying your nanoparticles. And in general, they're very fast. And so we leverage these high throughput plate readers that have already been developed for biological research. And many of them can take full spectra of an entire plate within the span of 20 minutes. So the first thing that we did when we developed Wanda was to try to understand how reproducible it was, because that's a really common problem in nanotechnology, is that many of the reports that you see are difficult to reproduce. So first I had my undergrads make 6 different manual syntheses of cadmium selenide nanocrystals. And you can see that they have this distinctive shape of a cadmium selenide nanocrystal. In particular, they have these peaks that tell you how big the nanoparticles are, and the width can tell you the distribution of sizes. So you can see that at a given time point, the spectra for the manually synthesized nanoparticles are fairly scattered because we don't have good control over the reaction. Whereas when we do these reactions in Wanda, at a given time point at 1200 seconds, you see that there looks like there's only one spectrum, but it's actually 8 spectra stacked on top of each other. So that means the nanoparticles that we made are exactly the same. And we didn't even normalize these arbitrarily. We normalized them only by the mass of the aliquots, as I showed you in the previous video. So Wanda is about 10 times more reproducible than a human, which is really important from a user facility standpoint, because a lot of times we get scientists who don't know how to make nanoparticles when they first come in. And it's also important for applications, because many times you want the same exact nanoparticle every single time. And even research-wise, you need to, when you're doing, for example, mechanistic studies with kinetics, you don't want very large error bars in your time points. So we've expanded our high throughput characterization capabilities beyond optical capabilities, including Raman, dynamic light scattering, X-ray diffraction, and even small angle X-ray diffraction and optical microscopy. So Wanda was our first robot, but we've since developed the second generation of robot, which we call Herman, which stands for the high throughput experimentation robot for multiplexed automation of nanochemistry. And the multiplex comes from the fact that instead of doing these reactions one at a time in an 8 reactor array, which isn't that high throughput, we in fact have a microplate reactor that can now react up to 96 different nanoparticles in parallel. So in addition, it has bells and whistles such as powder dispensing and viscous liquid dispensing. And importantly, it has an automated centrifuge. So this video is going to show you that we can actually centrifuge different microplates of vials. In this case, we don't have the vials in the video, but so I told you how optical characterization is really important and allows us to characterize our materials without purifying the nanoparticles. But for other types of characterization like X-ray diffraction and TEM, you do have to purify the particles. And this is really difficult for most new users because it's not typically well described in the literature. And also it can depend a lot on the different particle sizes and particle types. And so normally what we would do is that your reaction is this soup of nanoparticles and solvent and reagents. And so we add an anti-solvent to crash out the nanoparticles from solution. And after that, you need a centrifuge to make a pellet at the bottom of your vial so that you can decant all their supernatant. So having automated centrifuges on a robotic deck is really important for systematizing this really fussy purification process. So a third robotic workflow that we've developed is called RAPID, which stands for the Robot Accelerated Perovskite Investigation and Discovery Workflow. And this was developed in collaboration with users at Haverford University and Fordham, Josh Schreier, Alex Norquist, and also Sorel Friedler. And so they had developed this Escalate pipeline in conjunction with our RAPID workflow. And this pipeline generates experiments to run and also formats the data that it captures so that it's more amenable to machine learning. So in this case, we use a very standard Hamilton Nimbus liquid handling robot that can pipette stock solutions into this reactor block that can be heated and shaped. So then after in this case, we're making perovskite nanocrystals exactly like Farnaz described in her previous talk. And so the idea was that these precursors would make the macroscopic perovskite crystals, and then we want to see how big the crystals were, because normally for crystal structure determination, you want to have a very large single crystal. And so we can use optical microscopy and machine learning to classify the size of the crystals or the quality of the crystals. And then we can develop models that, for example, predict whether you get really nice single crystals, for example, in these red spots, based on the concentrations of your precursors. So if you want, you can feed that information for your model back into the escalate pipeline, which can generate new reactions to run that can update the model and make it more accurate. So this kind of sequential learning is often used in an active learning mode. So in active learning, you're updating your model to make it more accurate. And so in this case, we've used our rapid workflow and active learning to learn models that can predict the dimensionality of perovskites. So in this case, the dimensionality means not the size of the particle, but the connectivity of the octahedral building blocks of these lead halide perovskites. We've also used active learning to study the ligands that can enhance the luminescence of these perovskite nanoparticles. We also are studying meta-learning. In this case, meta-learning is learning how to learn. So you can train a bunch of machine learning models on different perovskites, and that's great. But the reality is that by the time you train a model, you really don't need to use the model because you've learned everything you need to learn about that perovskite. So what we really want to know is we want a model that can predict the optimal reaction conditions for a perovskite we've never made before. But when we tried to use our very large data set, so we trained a data set of over 17,000 perovskite reactions on 50 different perovskites. But when we tried to train a 51st perovskite, for example, or when we tried to predict the reaction conditions for a 51st perovskite, it was terrible, because machine learning models are typically not really good at extrapolating to parameter spaces that they haven't explored. So instead, what we can do is we can train a model that is good at learning new perovskites. In this case, instead of using, say, 100 perovskite reactions to train a new perovskite, we can say, let's just make 10 different random reactions and see which models can learn very quickly that new perovskite. And so after that, instead, after we use this, for example, meta-agnostic meta-learning model, we can then do active learning. So we can ask this model, based on these 10 reactions, what are 10 more reactions that you think it will allow you to learn even more? And so we used a variant of mammal called platypus, because of course a platypus is a mammal, to do active learning in this meta-learning mode. And so this is really exciting, because then it offers an ability to do a few shot modeling on these extrapolative problems instead of doing purely interpolation. So I've told you about 3 different robots. In fact, we have more than that. They're very powerful. They allow us to screen many different types of nanoparticles and do machine learning. This Nimbus robot is by far the most productive robot that I've heard of in terms of making colloidal nanoparticles. So you might ask, why do you need 4 or 5 different robots? Why don't you just use one robot? Well, the reason is that each robot has a different core competency. And this is really important, because you could make a robot that does everything, but it would be absurdly expensive and only one person could use it at a time. So instead, each of the robots plays a different role. For example, the Nimbus robots, which are really easy to use, can perform primary screening, where you're just trying to get the lay of a very large parameter space. Whereas Herman is in a glove box, it has more bells and whistles, and it can do this high throughput screening, but maybe in more detail and with more care. And finally, Wanda, which is larger but less high throughput, can make larger amounts of samples and do more in-depth characterization, for example, of kinetics that require higher precision. So one of the future capabilities that we're working on is automated TM, which was also referenced in previous talks. So a big gap is that when we make these nanoparticles, optical methods are great, but the optimal characterization is TEM, because they tell you exactly what the size and structure and defects of the particles are. Because even though we can just simulate them as a sphere or a cube, each of them really has an irregular shape and may have irregularities in its lattice. So the problem is that there are not a lot of high throughput TMs or automated TMs in the world. And one of the problems is that, when you're doing these analyses in TM, you're using a grid and you have to pump the grid into your TM every single sample. So instead, what we did is we purchased this side-TEM picoliter spotting robot that can spot many different samples on a single TM grid. And so therefore you can pump all these samples in a single grid and perform automated TM. And so our collaborators, Peter Urshus at the Foundry, are using very advanced TM, but tiling this frame across an entire grid and across each one of these samples. And so in this way, instead of having a compromise between high throughput and quality, they can actually perform these really exotic methods such as 4D STEM on every single frame in a TEM grid and calculate statistics instead of trying to image just a single hero particle. So I hope I've shown you today that robotic nanoparticle research is really powerful. So I'll leave you with this last vignette in which we use lanthanide-doped nanoparticles in our robots. And so in this case, the lanthanides control their optical properties. But the real value of these high throughput screens is that it allowed us to train physical models that then were used to predict something called thulium-doped energy looping particles. So this is a really unique nonlinear process that allows us to make continuous wave lasers and plasmonic lasers. And ultimately, these particles called photon avalanching particles that are some of the most nonlinear nanoparticles ever invented. And so this nonlinearity, which can exceed 200 now, so essentially like a 200 photon process, has allowed us to achieve two different modes of super resolution imaging, with confocal imaging getting down to less than 70 nanometers in resolution, and also localization imaging with less than an angstrom of localization accuracy. So I'd like to thank my group and also the Department of Energy and DARPA for funding this. And I'd like to remind you that the Foundry is a user facility. So if you'd like to learn more about high throughput and robotic nanoparticle synthesis, you can email me or go to this link here. Thanks.
Thank you. We will now go to the question and answer portion of our webinar. So I would encourage you, if you have a question, to put it in the chat. And we can just take a few questions. The first question, I guess, is for Nick. Is the Monarch facility open for users?
Yeah, the answer is yes, in a limited capacity. So we're in kind of the final throes of getting our 2D quantum material pipelines kind of fully developed and deployed. But our cryogenic characterization facilities, both at Montana State University as well as the University of Arkansas, are up and running. And so, yeah, we'd be happy to talk with potential users about their needs and needed timelines, and yeah, set up some joint projects.
Great, thank you. We have a question. Do you measure the reproducibility of particle number concentration, particles per milliliter? What kind of technique do you use? I'm assuming that's potentially Emory.
Yeah, so I can share my screen. So in this case, we do measure the particle number and the reproducibility. And that's one of the reasons why we need to weigh our aliquots, because we really need to know how much of each reaction aliquot came from which reaction. And so there are essentially calibration curves of extinction coefficients for particles of each size, in this case for cadmium selenide. These haven't been developed for every single nanoparticle, but for many of the popular ones, they have been. And so you can use, for example, the excitation or the extinction at 400 nanometers to calculate the overall amount of cadmium selenide you have. And if you know the size, then you can back calculate how many particles you have. It's still an approximation, but it's the closest that we can get.
Okay, thank you. There's a question for Naz on the perovskite and other array. I think I recall a distribution of performances through a much tighter distribution of the other samples. What do you think is causing the distribution of performance?
Yeah, that's correct. So for the nanoparticle array, based on our studies, majority of the distribution is still coming from the slight variation that remains in the particle size and shape as synthesized. So the remaining distribution largely is attributed to the variation that remains. So we do major filtering of them, but some remains. For the perovskite, yes, the variation is more, and that's because the variation optical response could come from variations in the perovskites themselves, size, structure, and all that as they're formed in an array. But those materials are also heavily influenced by the surface states, and the particular example I showed you are not optimized for passivation. So with surface passivation and optimizing the surface, we believe that will be reduced.
Thank you. And then our final question, what technique is used in the automatic TEM? It says STEM. I think.
So I believe that question would be for me.
Yep.
I think you can actually do various modes in the automated TEM. Some of these automated software programs are actually open source, but in this case, the Foundry or National Center for Electron Microscopy tends to use STEM a lot. One is, I think, for nanoparticles, the contrast is better. And also they've developed a whole suite of techniques such as 4D STEM or differential phase contrast STEM that are very useful for low contrast and for extracting out useful and quantitative information. So it doesn't have to be STEM, but it tends to be STEM a lot.
Right, thank you. And I'm going to invite Dr. Brandon Brow, the director of the National Nanotechnology Coordination Office, to join us for some closing remarks.
Yeah, thanks, Elizabeth. I know that we're over time already. I did want to say thank you and also a bit of a preview of coming attractions. So first off, thanks to the 3 outstanding speakers today, but also the speakers throughout this entire series. And thank you to Elizabeth, your moderation and your leadership, but also the leadership of many of the feds that have helped conceptualize and execute this event. I wanted to say that we've learned a lot about different ways that nanometrology is impacting kind of the cutting edge of science. And I think a lot of these techniques and tools have been new to us, but also there are constantly every day new techniques and tools being developed. And so in the next iteration of this webinar series, we're going to look into some of the people and some of their work in developing those new cutting edge tools and techniques, primarily those that are available at user facilities so that the audience can learn about what's on that cutting edge, but also not just kind of from an academic standpoint, but learn about what might be available to them that they can apply to their own research. So thank you to everyone. Thank you to the audience, and stay tuned. We will be sending more information in terms of what the next steps are going to be, but we look forward to joining you in more really great conversations. So thanks all.